Next, I am going to present you all our SCA Evolution Software, which is our open system. This is, you can see it in the screen, it's composed mainly by three components: a camera, which is a high-resolution camera from a German brand, connected to a microscope which needs to fulfill different requirements, and then a computer. After, some other optional components can be added. For example, a printer, label printer, a barcode reader, a fluorescence module, and other components. But at least you need to have these three. It's also possible to check the minimum requirements that these three hardware components need to accomplish, and then maybe some components that you already have in your laboratory can accomplish these requirements and you can use them. Also, it's important to know that this software, this system, is modular, in the sense that it's not necessary to have all of the analysis modules at the same time, but you can have just one for the moment, or a couple of them. For example, motility and concentration module and morphology, and then in the future incorporate other ones. So the available analysis modules that there are, the automatic modules, are motility and concentration analysis, which will be done at the same time, morphology, DNA fragmentation, vitality, Ackerson reaction, and leukocytes. So this is the main information about the system. It's different from our closed system, which is the most recent one, which is called SCA Scope. You see that they are quite different, at least in the hardware type, because this is an all-in-one system which contains a motorized worm stage inside, and that we can add up to 4 slides at the same time and perform the different analyses. This SCA scope has no fluorescence, so the models that are available here are the ones that can be run in the routine of a laboratory. So these are the models that are available, and this is more automatic. But today we are going to focus on SCA Evolution. So for this, I will open the software and make you a demonstration of the software. So this is the SCA Evolution software. This is the first screen that you can find, and this screen is allowing you to choose the user that is going to work with the system. By default, there are 3 users created that have, for the moment, different rights upon the system. For example, the administrator will be the one able to create samples, create analysis, run them, delete, change configurations. So they will be able to do everything. This will be different from the analyst, for example, that has just some right during analysis process, but after there will be some actions not allowed for the analyst. And the last one is the viewer, just able to see the already performed analysis and have the reports. It's possible to create and to customize this user window according to the people that are going to use the system in each center. And we can assign a password. Also from here, it's possible to choose the language. And then it's another important subject regarding SCA Evolution. It's to understand that there are different system types and different editions. System types mean that normally the system that is composed by the camera, the computer, and the microscope, it's called a full system. That in centers in which they just have one SCA, they will have full one. But it's possible to have different combinations of the software in case that there is more than one system in one center, or there are different clinics belonging to the same group that they want to share information. Maybe if in some clinic they just they just receive the images and the samples, and then they won't do the analysis right there. It's possible to have a capture system This capture system will include also the camera and the computer, but it will be cheaper because the analysis won't be done here, but in the full system that will be linked with this capture system. So it's just a capture station. Also, there is another possibility, which is the editor. The editor system won't require a camera because in this case the analysis will be open and then manual modifications could be done, checking the analysis, but just already analyzed images. And finally, the viewer. The viewer can be installed in any computer in order to check the already performed analysis without any modification. It won't be possible to perform any modification, but just review the different analyses. For example, the doctor could do this and print the report. On the other hand, there are different editions about the SCA Evolution software. In this case, I am going to show you human, but it's possible to have other ones. For example, for veterinary is for all of the species except rat and mouse. Toxicology edition is specifically for rat and mouse samples. Production, it's very focused on production centers, only working with one species and wanting to have just the main information about the analysis, and research with which we can analyze both human and all kinds of animal samples. So for the moment I will log in as administrator and we arrive to the main screen, which is the new sample creation screen. So here what you can see. In this case, the first thing normal to do when we receive a sample is to create a new sample into the software. Here there are two possibilities. Maybe we need to create all of the samples from here, from the software. So at the beginning of the day, all of the samples can be created, and then every time that we want to analyze each of them, just select the sample and then begin the analysis. But there is another possibility. Maybe it's possible to connect the software with the laboratory information system. In this case, all of the samples will appear here in the right part of the screen, and then it will be required just to select the sample that we want to analyze and start the analysis. So there are these two different possibilities. In my case, I will show you how to create a new sample and a new patient. You see here that there is a reference by default. This is the same reference that was created with the last sample with 1 unit more. This is to avoid sample duplicates. The reference of the sample needs to be unique for each sample. It's not possible to repeat. So this number will be automatically always different, but it's possible also to change it. So important, the sample reference is linked to the sample that we are about to analyze. In this case, I can introduce, for example, today's date, and then just like this, and it can be sample number 1, for example. So once that we have completed the reference of the sample, then we need to link the clinical history number for the patient. In this case, we can click here on search patient, and then this window will be open with the patients that are already in the database. So in this case, we can select a patient because maybe 3 months ago we already analyzed a sample from this patient, and now we have again another sample, so at the end it will be possible to compare these two samples. If not, we can just create a new patient. And when creating a new patient, it's possible to add different information, but at least in order to save the patient, we need to fulfill two informations, which is the clinical history number of the patient. That can be a number that each center assigns. For example, I will go again with today's date and the first name. for example, George. Other informations, for example, the last name, the email, the birth date, the language of the report, and other optionals can be also fulfilled by the patient, for the patient. So here the purpose is to have all of the information about the patient collected, because it will be possible to transfer this information to the final report. So for example, if you want to add the telephone number of the patient or the wife's name or previous diseases, it's possible by double-clicking to change this word. And for example, phone, change this, and also change this, and this will be changed for all of the patients in order to have homogeneous information. And for example, previous diseases. And then this could be fulfilled with information that you want. So once that we have finished, we can validate. We have our patient here, and we will select it. So now that we have assigned to the sample a reference number and a patient, we move on to the macroscopic and microscopic data for the sample. What we find here are a set of parameters. This set of parameters are defined by a WHO manual and are very important to take into account for after interpreting the results of the different semen assessments, and then taking everything into consideration, arrive to the final diagnostic. So, for example, we have here when the sample was delivered, when the analysis was begun, the collection date, the volume, very important, the volume of the sample, because the concentration will be calculated in million per milliliter, but also in total million per sample. The pH, also the temperature at which we are working, the abstinence days, and then a set of other parameters, for example, if there have been collection difficulties, if the liquefaction is complete or not, the sample is viscous, if there are aggregations. So at the moment when we create the sample, maybe it's not possible to know if there are aggregations or agglutinations. But we can always come back to this screen once we have run the motility and concentration analysis, and then just complete the information that was missing at the first moment. And also, you see here that there are 14 optional fields that can be used to add additional information, this time about the sample. So you see that I have already changed some of these parameters. The technique is the same. If we double click here, or the same, we click right, we can change the title, we can change the word, and then add other information that can be either qualitative. For example, if we change this and we want to introduce the color of the sample, we can here introduce white, for example. So this would be qualitative information. Or quantitative. If, for example, you perform additional manual tests that are not automatic, in this case it's possible to perform them in the microscope and then add the results here. So this can be done, for example, if you perform the ROS test, the oxidative stress test, or the antibody anti-sperm test, for example, EGA. You can directly introduce here the result of this test by writing, for example, this number. Or if you double click, a manual counter will be open, and you will be able to use the 0 and the dot in the keyboard to count positive sperms and negative sperms concerning the IgA test, and then link that in this field we are assessing EGA. So if you count different cells in the microscope, you can use the keyboard of the computer as a counter, and then save, and you will have here the results and the units. So this can be used for additional tests, manual tests, and then all of this will be transferred in the report. So once that we have completed the macroscopic information, we can move on to microscopic data, microscopic analysis. So here we open programs, and then we begin with the first analysis recommended to do during the first hour after ejaculation, which is motility and concentration that will be performed at the same time. So I click here in motility, and then as you see in each module, analysis module, it's possible to create different configurations in case that we analyze samples at different conditions. For example, parameters for a fresh sample, fresh ejaculate, won't be the same that for capacitated sperms, or for example, if we perform motility in underfluorescence. So it's possible to adapt, just to click and begin the analysis. Also, here, if you have introduced the volume, here the volume will be transferred, and it's possible to introduce a dilution factor. In this case, it's important to take into account that the software will analyze properly samples between 1 and 100 million per milliliter of sperms. With samples that have a concentration under 1 million per milliliter, maybe there are some centers that directly do the analysis manually. They concentrate the sample and follow the guidelines of WHO. And for samples having a concentration higher than 100 million per milliliter, in this case, it's recommended to dilute the sample. And this is a message that you will obtain through the software. When you get a higher concentration, higher than 100 million per milliliter, the software will show a message advising to dilute. And to dilute, you can use seminal plasma or a saline solution or other media that you have available. In this case, the next time we will dilute a fraction, an aliquot of the sample, just to do the motility and concentration analysis. So in this case, you can dilute the double, or well, a half or a third of the sample. And the way to introduce the dilution factor inside this module is to think about it as a ratio. Normally 1,0 means 1 part of sample and any part of diluent. But 1,1 means that we have mixed 1 part of sample and 1 part of diluent. For example, that we have mixed 10 microliters of sample and 10 microliters of seminal plasma. And the same thing. If we put 1,5, this could mean that we have used 10 microliters of sample and 50 microliters of diluent. So this would work as a proportion. Having said this, we can also keep talking about motility and concentration analysis. In this case, here you have the configuration, and we will work in positive phase contrast and with a 10 times objective. And here in the configuration, for example, you have the parameters that are followed during this analysis, which are the ones given by WHO manual, but it's possible to change them. So if we click here, Analyze, we will need to load our counting chamber, put it in the microscope, focus, adjust the light, and then begin to capture. So for capturing, we need to keep clicking Analyze. So we can click Analyze or the spacebar in the keyboard, and here we can see the progression of the fields and the number of fields and the number of sperms that we have analyzed. In this case, it's recommended to analyze at least 3 fields or 200 sperms. But of course, as much sperms are counted, the better will be the analysis and the more accurate will be the analysis. In this case, we have analyzed 360 sperms in 5 fields, and the software asks us if we want to see the final results. So we can click on yes, and we will arrive to the screen of the results. So here, first of all, before checking the results, what we can do is to check the homogeneity of the sample. If we go to Comparison tab, we can test, we can check the color of the videos that have been captured. In this case, there are 2 of them which are red, meaning that they are heterogeneous. They have a variation superior to 20% in comparison with the average of the other fields. In this case, we could open the field to check what has happened, if there is some bubble or there is an aggregation or some kind of problem. And if you see a big problem in the field, with the right click, we can just delete this field. If not, if for example the sample is viscous, maybe we can assume that it will be heterogeneous and we can keep working like this. And also, before going deeper into the results, we can, with right click, view the field. And these are the kind of videos, the 1-second videos that will be recorded during motility and concentration analysis. Here you can see the type of image that we expect to find when using positive phase contrast. These are the sperms with white heads, the background is gray, and then there are some particles in the background that may be artifacts and that we are not interested in black. So just the sperm heads will be analyzed. And you see that there are different colors for each sperm. This is because now we are seeing the WHO 5th edition classification into 3 categories: progressive, the red ones, non-progressive, and immotile. It's also possible to select WHO 4th edition into 4 categories: rapid progressive, type A, type B, C, or D. So you can check this. This classification will be the same as in WHO 6th edition. And in fact, nowadays we are in process of adapting the software to the new version of the manual. So we will come back to this classification. Also, it's possible from this screen to count the concentration of round cells. Round cells are all these kind of cells that are not sperms and can be found in semen sample. That can be, for example, germinal cells or leukocytes. So these are the kind of round cells. For example, this one or this one. In this case, for the moment, we cannot distinguish the origin. But after, if the concentration of round cells is higher than 1 million per milliliter, then WHO recommends to perform leukocytes analysis in order to discard a possible infection of the sample. Other thing important is that we can just double click on one sperm in order to see the individual information about this sperm, all of the velocities. These are the curvilinear velocity, the average velocity, straight line. So these are individual information about this sperm. And you see that the video, the analysis are automatic, but it's possible also to perform manual modifications. Here in the left part of the screen, there is a toolbox that can be used to add or delete some sperms. So if you want to improve your analysis, it's possible. If you see that some sperm has not been analyzed or due to reasons of focus or contrast, for example, this one here in the bottom, this would be an immotile. So you can click here in Add Sperm manually and add a type D. And every time that you do modifications, the results are recalculated automatically. So you can do some modifications or even delete something that it's not an sperm by clicking here in the cross and clicking here in the screen, for example. Also, it's possible with the right click to select areas in order to delete the analysis. This can be used, for example, if you are facing to an aggregation or other type of problems during analysis. You can definitely try to remove one big area or undo your changes. So going back again to the results, we can check different results. On the one hand, the concentration in million per milliliter and in million per sample. Also, the different percentages of motility and progressivity. And here we have the 4 categories for the motility, type A, B, C or D. And here in the left part of the screen, for the moment, we have the diagnostic of our sample. If we want to have more information, we can go here to results 2 and check some functional tests that can be also calculated during the motility and concentration module, which is, for example, mucus penetration. Here, mucus penetration is a label, it's a subpopulation of sperms that the software can calculate an approximation of how many sperms from the sample in physiological conditions could be able to cross cervical mucus in female tract. These sperms will be selected by the way that they swim, the speed, and if they are moving forward or not, if they are progressive or not. In this case, we will have the approximation, the calculation, and we will have here the results of, for example, in this case, 6 million, almost 7 million of sperms in this sample are correct, would be able in physiological conditions to cross cervical mucus. This can also help us to choose the technique for the insemination. Also, hyperactivation can be studied. In this case, in a fresh sample, it doesn't make sense to test hyperactivation because normally it's 0% in an ejaculate, but in a post-treated sample or a sample that we have incubated with an hyperactivation media, it's possible to check the percentage of hyperactivation based on the high speed that the sperms have when they are hyperactivated and also on the star-like movement that they adopt. So in this case, it would be possible also to test the functionality of the sperms, if they are able to be hyperactivated or not. And finally, also the concentration of round cells. In this case, it's important this value, the concentration in million per milliliter of round cells. Here it's almost 1, which could be the cutoff to continue studying the sample to differentiate the origin of the problem. Next, there are some parameters linked to the kinematic of the sample. This is more focused on the research, but you have the average of the velocity, the curvilinear velocity, the average velocity, the laterality of the head, and other values. And the complete list of sperm that have been captured during this analysis. This table can be exported to an Excel file by obtaining the report here, and when clicking here in spermatozoa, this table will be automatically exported in an Excel file. If not, it's also possible to create our own table, selecting just the columns and the parameters that we are interested in. In this case, it will be also possible to create, to export this data to an Excel file. So this is concerning the motility and concentration analysis. So now we will save and close. Every time that we close an analysis, it will be automatically linked to our sample, and all of the videos will be automatically included and saved in the computer. Moving on to the next analysis, which is chronologically vitality. Vitality is also a time-dependent analysis, and it's recommended by WHO to perform it during the first hour after ejaculation. So, vitality is not mandatory for all of the samples, just for those samples with low motility. Why? Because we could be interested in knowing if the immotile sperms that we find in our sample are alive or are dead. In this case, we could be facing two very different problems, if they are dead or if they are just immotile. Maybe because there is some problem during the motility acquisition, during spermatogenesis, or if they are dead, they may be linked with other problems. For vitality, the kit that we suggest is a solution which is called Brightbit, which is a solution based on eosin nigrosin recommended by WHO. This analysis can also be done under fluorescence, but normally with the Brightbit it's easier to do it with using bright field in the microscope. So all of the modules, you will see that they work the same. So in this case, we can go again to analyze and capture some images. In this case, at least 200 sperms would be recommended. So I will capture some of the images. Of course, here the technician needs to prepare the sample and then put the sample in the microscope and focus it. So once that we have captured some images, we can go again to the results and it will be the same thing. If we view this field, you see that here there are some sperms with a green square or with a red square. These are classified as alive and the other ones are classified as dead. So this is the difference. These colors can be changed. Sometimes alive sperms can be in blue and dead sperms in red also. And here with this solution, what we are testing for is the membrane integrity. So eosin solution, eosin will just enter to the head of those sperms that are dead, and eosin won't be able to enter to the heads of a live sperm. So for this, you can test for the difference. Of course, also we have here the toolbox that can be used to do manual modifications. And finally, find the results of alive and dead sperms. So I will also save and close. We move to the next analysis, which is morphology. For morphology, different stainings are compatible with the software. DivQuick, Papa Nicolau, and others that I can show you. DivQuick, Papa Nicolau, Shore, CellView, Sperm Blue, Sperm Stain, and other ones. And also it's possible to choose different criteria for morphology analysis. We can choose the WHO, Kruger, or the bit modifier, or even we can create our own normal sperm values, ranges, and we can customize this screen. But now we are going to leave WHO and then begin the analysis. In this case, the technician would need to prepare the morphology smears and then stain them and then just begin to analyze. The morphology analysis can be done, as we are using a high resolution camera, can be done with a 60 times objective at bright field. For vitality, I haven't said that we can use 10 or 20 times objective, but for morphology 60 times. And we recommend to use mounting media, like for example EUKIT, because even though we are not working at 100 times objective, still we are working at high magnification, and then always the focus and the coloration are easy to do with a mounted slide. So I can capture some sperms and then go back to the results. So for morphology, you see that some sperms are classified as abnormal and others as normal. And if we double click on one sperm, we can see why this sperm has been classified as abnormal, because of these parameters and then also the classification that the software has given to this sperm. So it's possible also to do manual modifications. and you see that here the acrosome it's yellow, the post-acrosome it's blue, there is also the midpiece and the tail. So all of these structures are taken into account during the analysis. And this is the kind of coloration that sperm blue staining provides. You see that there is a good contrast between the acrosome and the post-acrosome, and other structures can be also studied. To facilitate things and if manual modifications are required, there is this window, which is edit window, in which we obtain the complete list of sperms analyzed during morphology analysis, and it's possible to filter by abnormality index, having at the top of the list the most normal sperms and at the bottom the most abnormal ones, with more parameters abnormal. In this case, we can from here modify, do manual modifications, results being recalculated, and also delete some sperms, or delete multiple ones if they are not sperms. Regarding the results, as you can see, the results are expressed like the following: percentage of normal sperms, percentage of abnormal sperms, and then all of the abnormalities counted in general. So in this case, there are 60% of sperms with head defects, 40% with midpiece defects, also tail defects, and cytoplasmic droplets. But if we want to know more, we can go also to this screen sort, in which 3 indexes are calculated. The 3 indexes recommended by WHO, which is the teratospermy index, multiple abnormalities index, and deformity index. So this can of course be taken into account, not just to stay with the percentage of normal sperms, but also to know more about the abnormal fraction of the sample. In morphology, we have more detailed the abnormalities of all of the sperms. This is according to WHO, but again, the criteria can be selected, but can be chosen. And the main values for morphometry. In this case, this can be used more in research, not that in routinary analysis, but in research this can be used to publish some studies and compare different types of samples. And also we have the advanced option here. So I will save and close again. We can move on to the next analysis module, which is DNA fragmentation. In this case, DNA fragmentation is studied thanks to the technique called sperm chromatin dispersion technique, or also the halo test. In this case, we are going to incubate our sample with different solutions that will produce that intact correct sperms will generate a big halo around the heads, and in contrast, damaged sperms will not generate any halo. And then we will stain the slides in order to appreciate the halos. So if we go here to analyze, we are going to do this analysis also with 10 or 20 times objective and in bright field. And the software will take into account the halo area to classify each sperm on fragmented or non-fragmented. You see, this is for example a fragmented small halo sperm, but all of these are non-fragmented. You see around the heads. Fragmentation analysis, it's not a basic routinary analysis, but when maybe the other sperm assessments, the other sperm parameters are normal, in some cases the problem could be linked with DNA fragmentation, not just in problems related with fertilization, but also with embryo development. So this is a more advanced analysis, but that in the last years it has gained some importance, and more clinics are running this test lately. So this is a kind of image that you can get, and when clicking on one sperm, we can see the individual information about the halo area and the classification. So we can take a look at the percentage of not fragmented and fragmented results, sperms, and also the number by the halo. For example, big halo or medium halo, small, without, or degraded. So I will also save and close. We move on to the next analysis, which is Ackerson reaction. For the Ackerson reaction, we need fluorescence. This is an additional module, and because this can be only run with the use of fluorescence, and two fluorochromes, which are Hodge and PNA. PNA is a fluorescence dye that is specific for the Ackerson. So we will see two types of sperms here. Some sperms with intact Ackerson, which will have this kind of coloration, green thanks to the PNA and blue for the rest of the Hodge, and other ones with exploded Ackerson, reacted Ackerson, just blue. So we will study a control situation, the ejaculate, capacitated, and then we can save these results and then do again the Ackerson reaction analysis, but with a sample that normally is incubated with calcium ionophore in order to force Ackerson reaction. In this case, this is a treated sample, this is a sample of a study, and we can capture other images, for example, these ones. And in this case, we normally expect to find more sperms without Ackerson because we have incubated them with calcium ionophore, which forces this Ackerson reaction. So if we save the images, then if we go here to Ackerson, we can check the difference between the two conditions, control condition and the reacted, the sample incubated with calcium. And this difference is expected to be higher than 15%. If not, we can then test for the different origin problems. And finally, we are going to talk about the leukocytes analysis. Leukocytes analysis is also not mandatory, and it's recommended to be performed during also the first hour after ejaculation to discard the presence of leukocytes in the sample. So in this case, this test is done at bright field and with the objective of 40 times. And the sample needs to be stained with a substrate that it's prepared, a fresh substrate, and then just the peroxidase positive samples, meaning the leukocytes, will be stained in dark brown. The negatives will be without color. These without color, for example, these ones will be germinal cells. They are peroxidase negative, but the leukocytes will be dark brown, will be positive. So at the end, we will have the concentration of peroxidase positive cells, leukocyte cells, and if this concentration is higher than 1 million per milliliter, then it would be not recommended to use this sample for art techniques. So we can save and close. And finally, just to mention that there is a manual counter, which is not an automatic module, but it's a manual counter that provides us with the images that we can capture, but it works the same as the other modules, but here the classification is manual. So we can run any analysis with this manual counter, but we would need to do the classification. Okay. And finally, the final step after performing a complete analysis that I have shown you is obtaining the reports. By default, there are a lot of reports that SCA Evolution provides us with. There is, for example, a summary report with the main results of the analysis. And it's it's possible to export this report in PDF, in Word, or to directly print the report. Or also it's possible to obtain a complete report containing a lot of information, also some graphics, some images, the macroscopic information about the patient, and all of this. And it's also possible to export in Excel, in Word, in PDF, or other formats. Imagine that you have seen also the validation option. It's possible to enable the validation in the cases in the hospitals in which there are some laboratory technicians, and then some biologist or someone else needs to check the analysis before sending the report, the final report, to the patient. Also to mention that here in Microptic we count with strong technical support during our working hours. You can also you can just ask for remote support and we will help you by seeing your screen and taking the control of the mouse. So if any error message in the screen or any problem or doubt, it's possible to ask for remote support, and online in the distance we will help you, and the majority of the problems can be solved like this. Okay, so I hope you have enjoyed this demo and thank you for your attention.